Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on enzymology of DNA replication. In DNA replication, so many enzymes, they work together to accurately copy the DNA molecule. Let's see what are the enzymes involved in DNA replication first. After that, I will explain each and every enzyme. Okay, so let's see. First enzyme is Helicase. Second one is single stranded binding proteins. Or SSB. Third one is Gyrase. Fourth one is RNA Polymerase or primase. So fifth one is DNA Polymerase 1. Sixth one is DNA Polymerase 2. Seventh one is DNA Polymerase 3. Eighth one is Ligase. Ninth one is RNase H enzyme. It is also called as ribonuclease H enzyme. 10th one is sliding clamp. It is also called as beta clamp. So let's see the first enzyme, that is helicase. So helicase binds at origin region. So it separates the two DNA strands by breaking the hydrogen bonds present between the two strands. Okay, so it moves along the replication fork by utilizing ATP. So it creates the replication fork. Okay, so it separates two DNA strands. The separation of two DNA strands is nothing but melting. Okay, so it also works together with SSB. SSB nothing but single strand binding proteins. So SSB, they keeps the two strands apart until the new DNA strand synthesized. Okay. So let's see the second enzymes, they are nothing but SSB. SSB is nothing but single stranded binding proteins. So right after the helicase unwinds the DNA duplex, the single stranded binding proteins will come. They attaches to the single strands. They attaches to the DNA single strands. Okay. Actually, they prevents the rejoining of these two strands until the new DNA is synthesized. Okay. They also prevents the formation of secondary structures. Okay. So let's see the third enzyme, that is nothing but gyrase. This is also called as topoisomerase. So gyrase is a kind of topoisomerase enzyme. So it is found in prokaryotes. For example, Escherichia coli. It relieves the supercoiling. Supercoiling is nothing but extra twisting or extra coiling. So when helicase is doing the separation of two strands, so it leads to the tension creation in between two strands that causes supercoiling. So that is relieved by the enzyme gyrase. Okay. It also helps in DNA replication continue smoothly. So that's why this enzyme is also called as tension releaser enzyme. Okay. So let's see the next one, that is primase. So primase is also called as RNA polymerase. So after unwinding of two strands of DNA, the bases present at the two strands, they are exposed to the enzymatic activity. So initially, the RNA polymerase will come and bind to the one strand. That strand acts as a template strand, you should remember. The strand direction is 3' to 5' direction. Okay, so it binds here. RNA polymerase binds here. So it will start the transcription. Okay, it starts the transcription. Then, so here 10 to 60 nucleotide length RNA transcript will be transcribed. So RNA fragment will be transcribed. 10 to 60 nucleotides length RNA fragment will be synthesized. So this is called primer. So it gives the initial step for the DNA synthesis. Okay, initially RNA is synthesized in the replication. You have to remember this. Okay, so RNA primer is having 3' OH group free. So now the DNA polymerases will come and they add nucleotides one by one to the 3' OH free 3' OH group. So now one nucleotide will come and join to this nucleotide. Okay, so resulting in the formation of phosphodiester, there is an establishment of phosphodiester bond between the nucleotides. So in this way the chain is going to elongate it. Okay, so let's see the next important enzyme, that is nothing but DNA polymerase 3. So it is a main enzyme which is responsible for the synthesis of DNA. So after the synthesis of primer, now RNA polymerase 3 will come. It binds here. Okay, so it adds nucleotides one by one to the growing chain. So it requires free 3' OH group of the previous nucleotide. Then only it will add one by one nucleotides to the growing chain. So okay, it requires free 3' OH group. Then only it will add one by one nucleotides to the growing chain. Okay, so the template strand direction is 3' to 5', whereas the newly synthesized strand direction is 5' to 3'. Okay, so it moves towards the replication fork. Okay, helicase unwinds in this way. Okay, so this strand moves continuously towards the replication fork. This strand is called as leading strand. Okay, so after some time, the synthesis of next strand occurs with the help of RNA polymerase first and then DNA polymerase next. Okay, so here discontinuous fragments are synthesized after they rejoin together with the help of an enzyme ligase. So this strand synthesis occurs discontinuously. That's why this strand is called as lagging strand. Okay. So DNA polymerase 3 can do the polymerase activity in 5' to 3' end. Okay, in 5' to 3' end. This is the 3' end, this is the 5' end. Okay, it can also do exonuclease activity in 3' to 5' direction. Okay, it also consists 3' to 5' direction exonuclease activity. Exonuclease activity is nothing but it can remove the unwanted nucleotides at a time, okay, from the ends of DNA strand. That is nothing but exonuclease activity. It can do polymerase activity for both the leading strand and also lagging strand. You should remember this, okay? So let's see the next enzyme, that is nothing but DNA polymerase 2. Okay. Actually, DNA polymerase 2 is involved in SOS DNA repair. SOS repair is nothing but when the DNA get damaged. So, this can overcome the damaged DNA and allows the DNA to continue the replication. So, that is nothing but SOS repair. Okay. It can also synthesize DNA strand in the 5' to 3' direction. That means it has the polymerase activity in 5' to 3' direction. It also have the proofreading activity in the 5' to 3' direction. So it can check and correct the mistakes that occur during replication. That is nothing but proofreading activity. DNA polymerase 2 enzyme is also called as backup enzyme. Why? Because when the DNA polymerase 3 get damaged or the activity is blocked, then DNA polymerase 2 can take over the responsibility of DNA replication. That's why DNA polymerase 2 enzyme is also called as a backup enzyme. Okay. But it does not have the exonuclease activity. It have the polymerase activity and also proofreading activity, but it doesn't have the exonuclease activity. Okay, so let's see the next enzyme, that is DNA polymerase 1. In E. coli, DNA polymerase 1, it removes the RNA primers by its exonuclease activity. Okay, so it removes the RNA primers by its exonuclease activity. Okay, it also fills the primers gap by doing polymerase activity. There it synthesizes DNA to replace the gap. Okay, so DNA polymerase 1, it also have proofreading activity. Proofreading is nothing but it corrects the mismatch bases. It removes the wrong nucleotides. That is nothing but proofreading activity. So DNA polymerase 1 have polymerase activity, exonuclease activity and also proofreading activity. Actually, these three enzymes were reported in E. coli by Korenberg and team. So these three enzymes are DNA dependent polymerases. So let's see the next enzyme, DNA ligase. So DNA ligase, it joins the Okazaki fragments. So DNA ligase, it joins the Okazaki fragments by establishing phosphodiester bonds in between the nucleotides. Okay, the gaps were filled by ligase enzyme. The gaps present between Okazaki fragments were filled by ligase enzyme. Okay, so it establishes the continuous strand. Okay, so let's see the next enzyme, RNase H enzyme. This is also called as ribonuclease H enzyme. Okay, so it is responsible for the removal of primers in eukaryotes. So RNA polymerase 1 can remove the primers in E. coli prokaryotes, whereas RNase enzyme that removes primers in eukaryotes. Okay, so actually it works together with the DNA polymerase delta enzyme to replace the RNA with DNA. So the RNA present in the primer, that place is replaced by DNA with the help of an enzyme, RNase H enzyme. So let's see the last one, that is nothing but sliding clamp. or beta clamp. So, sliding clamp is a ring-shaped protein. So, actually, it holds the DNA polymerase on DNA. Okay. So, it is a ring-shaped protein. It holds the DNA polymerase on DNA. So, in prokaryotes, it is called as a beta clamp. But in the case of eukaryotes, it is called as pCNA. pCNA is nothing but proliferating cell nuclear antigen. Okay? Okay, students. This is about the enzymology of DNA replication. If you like this video, please share this video to your friends and subscribe my channel. Thank you so much.